Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Spring 2019 National Meeting in Orlando. We're joined today by Dr. Jonathan Barnes of Washington University in St. Louis. He's studying a muscle-like material that expands and contracts in response to light. Dr. Barnes.
Thank you very much, Sophie. Thank you guys for the opportunity to be able to hold a press conference. It's really nice for scientists to be able to discuss their research. I know that I'm going to sidetrack here with a little bit of a story, but in the U.S., scientists are not treated like celebrities, so this is a rare occurrence. And I remember when I went to Stockholm with my PhD advisor when he won the Nobel Prize, and people were just rushing him to like get his autograph, and it was so weird to see that, to have people like admire scientists. I think it's really cool that you guys are doing this. Okay, so we'll jump right in. Sorry, I'm Mr. Tangent. So like Sophie was saying, my lab has actually developed a new type of redox responsive polymer, and this polymer has the ability to accept electrons and to be able to fold and contract and make objects change their shape and mechanical properties. So let me advance the slides here. Where is the cursor? There it is. Okay, so as you can see here, this is one of my favorite images, is an elephant playing basketball, shooting free throws, and boy, could the Lakers use this elephant right now. They're officially out of the playoffs, if you were wondering at home. And what's really fascinating about this is a very large animal, right? And it has the ability to access certain features and muscles to be able to lift such a large object and to have changes in mechanical properties such that it can balance itself on its hind legs. And then likewise, you know, you've got the governor, Arnold Schwarzenegger here. We could debate whether that is natural actuation in terms of being able to change shape and size, or unnatural means. But nonetheless, when he flexes his arm, that muscle that's extended then becomes contracted and changes its mechanical properties, becomes stiffer, right? And then if you want to think about a smaller scale, you want to think about plants just kind of responding to visible light, then you can think about trees or plants that bend towards light in order to undergo the photosynthetic process. And so these are forms of natural examples of actuation, and so we're interested in mimicking this. And so thinking about you know, the makeup of what human muscle is comprised of, it's actin and myosin filaments that glide past each other, and you get about a 27% contraction in the one dimension, in the linear dimension. And so so this is fascinating because we're able to utilize this, and we're able to grow these and do self-healing and build muscle and make these even increase the performance of these materials, so-called materials. And so this is something that was really intriguing to me, you know, as a graduate student not that long ago. And so I started thinking about polymers that look like the one that's on the slide right now, where you have a linear chain that has these individual little units in it. And the question is, can we get that chain to contract like an accordion and expand when we want it to, in response to some electrical or light or chemical stimulus? And so this is actually, I always like to point this out, this is actually a slide that I use to interview for faculty positions. I have not changed it to this day because this was a big what if, right? This was a big question. Is this possible to make a polymer that looks like this one down here in the bottom, that is bottom right, that is blue and has all the positive charges? And then can we add electrons into each of those blue subunits and then make them look like the ones on the left? So I guess you guys can't probably see the cursor that I'm moving here, but have them reduce, and so you lose positive charge, and then they like to stack on top of each other. And so then the fundamental question is, okay, if we can make that type of polymer, then can we incorporate that into 3D polymer networks, so hydrogels and things of that sort? And then can we activate the polymer such that it tugs down on the rest of the polymer network and causes it to contract and change shape and become stiffer and more robust, very much like a muscle, right? That's what we're going for here. So I skipped through a lot of the steps to synthesize the polymer to save you guys the time. But what we actually did is we made hydrogel. So what you're looking at now is a hydrogel that is cubic or disc-like in shape. And this is actually when we use a chemical reductant, you can actually get these to shrink down to a tenth of their original size whilst being submerged in water. So these things can do this underwater. And in so doing, our polymers are actually what is called electrochromic polymers. So that means when you put electrons in, not only does it contract, but it also changes colors. And so they actually become opaque or black like this. And so you may be familiar with this process if you have an auto-dimming mirror in your car. So when you have headlights that are really bright behind you, that starts to dim and it starts to go kind of a grayish or faded out, that's vialogens. That's the little molecule that we put into these polymers. They actually have that in between two conductive plates that serve as an electrode that can reduce them. And as they're reducing, they get darker. And so it's an auto-dimming feature in cars. They're also on airplanes now, too. You can just push a button. Instead of lowering the shade, it just starts to go black. And so that way you can block the sunlight. So they're electrochromic, but when you integrate them into a network, like we did, a hydrogel, you can actually get them to shrink and contract. And this is completely reversible. So when you swell it back up, it regains its original shape. It regains its original soft-like nature. So these hydrogels are really soft. It's like Jell-O. If you could just push your thumb through it, and it would just tear it apart. But when it's reduced and contracted, it's actually a lot more like a rubber band. It's much tougher. And so that's what we're going for, is to be able to introduce those new properties and then reverse them back. And so we showed that we could cycle it over several cycles. We actually we wanted to publish this one actually in the first journal that we published this in Chemistry Materials, but we weren't allowed to because it has our logo, so I can only show it in presentations. But it's really cool because what we're showing here is a disc-shaped gel that has none of our active polymer, and then the WU for Washington University actually has it. And so you can see in this little GIF, when we pour the chemically reducing solution on top, it starts to change colors pretty quickly, and within 60 seconds, it's already really black. And so if I were to continue this video, you would see it actually contracting. I like to show this example because I think it resonates a little bit better. You can literally put our polymers into any type of hydrogel, any shape, and any size, right? So this is something that we're going for, is being able to scale these materials. And the reason why we can do this is because very, very small amount of our polymers actually needed to go into, for example, the gummy bear gel and get it to shrink. And what we see is that the features of the gummy bear are still retained. So it's an isotropic contraction. It happens in all directions and dimensions. And so this is again completely reversible, and you can swell it back up in water, and you can oxidize it, and the color fades. And so we wanted to then see if we could actually control this process with light. And so what you're looking at is essentially a experiment where we put a hydrogel, we glued it to a piece of black electrical tape and hung it from the top of a jar, the lid of a jar. And then we shine blue light on that and actually get it to bend and contract. And what it's doing is it's actually lifting that little yellow object at the bottom is a sponge that weighs 20 milligrams. And so you might be looking at this and you say, well, that's wonderful. You lifted a 20 milligram sponge and it took you 4 hours to do it, right? But what's nice about this is that this is an autonomous process, right? We shine light on it, and this process occurs inside where we're now reducing the polymers, and they're starting to contract and bending this. And so in that hydrogel, it weighs 500 milligrams, and of that, only 20 milligrams are actually polymer, and of that 20 milligrams, only 0.7 milligrams are the active polymer that we made. So very, very little of our actual polymer is needed to get this process to work. And so that's what I meant by the scalability of this material. And so we did the controls too, where if you take out some of the light-absorbing photocatalysts, nothing happens, no contraction occurs. So we'll go past that. And so what we're doing now is those previous gels were kind of weak. Like I said, they're very soft. You could kind of destroy them very easily if you were just to pick them up. And so now we're switching the chemistry. We're trying to make these materials tougher and stronger, and we want them to contract faster, right, more quickly, because 5 hours is pretty slow, right, in terms of contraction. And so we're trying to now come up with ways to, let's see if that'll play. Yeah. So if we have a hydrogel here that's clamped at the bottom, you can't see it now because the light went off, but there's about 50, I think, grams of weight that are just dangling from the end of this gel. And we shine blue light on it, and the gel starts out looking like this one over here on the left, but then immediately you can see it starts to turn purple, and eventually it'll turn black. And so it starts contracting, and you can actually look at the position of the bottom clamp as it's rising up. And so this is a little bit, it's still relatively slow, but it's a lot faster. It's 3 times faster than what I showed you on the previous slide. And so again, we can now pick and choose whatever type of monomer that we want to use. So any monomer that's used to make normal manufactured goods, we can actually incorporate our polymers with those monomers. So it's very adaptable. Not only is it scalable, but it's adaptable. So whatever properties you want, we can actually put our polymer into that. Okay, so let's go, almost done. Yeah, so we decided to actually do a photo patterning experiment. So we made a photo pattern that looks like the US flag, and then we irradiated a gel, and so you can actually see, we can actually get precise resolution when we pattern these substrates. And what's really cool is the regions that are being patterned, the ones that change color, you can actually tell where it's irradiating and where it's changing, but in those regions, the mechanical stiffness is actually increasing by a factor of 2 to 3. And so this process now, what you're seeing is a gel that was patterned, and then we just set it out on the bench top and let it oxidize, and it completely reverses. And so you can photo pattern reversibly different gels with whatever pattern you want. And so in this paper that we currently have under review, we've just taken the same gel and just photo patterned it several times, and just it's reversible, the whole process. And so that's what we're trying to do, is we're trying to get to a greater level of control with these materials, and to make them tougher, stronger, faster. I know that sounds like a commercial. Maybe it is. So buy our polymers, eventually. No, but anyways, all right, I'm a goofball. I'll keep talking. Or I'll stop talking. How about that? Thank you guys so much. This is my wonderful group and the funding sources that we're so grateful for, and I'd be happy to answer any questions. So, oh, yes, sorry. I actually have samples. Thank you for that cue. So this is actually what the gel looks like. It's like I said, a kind of a very similar to, essentially similar to a contact lens, but it's a very thick contact lens, and so it's got some nice stretch. It's got some nice elasticity to it. And so we take, this is the original starting hydrogel, and then we photo reduce this. We shine blue light on it, and then after a certain amount of time, it looks like this. And so these are literally essentially the same exact 2 gels, but this one is now contracted, and the mechanical properties have changed. And so this one is, you can do, well, that one broke, but that's also because we cut it. But it's got stretchier abilities, and so you can actually feel the stiffness and the change in the properties of the gel. And then if you let it go all the way down to the final shape, it's this dark, kind of smaller object that's black. Again, these are photochromic, and it's a lot harder to stretch these and get them to expand. Whereas, again, if you go back to kind of the original starting state, these are bouncier and kind of like a contact lens. So it's again, this is something that we've adopted just to make them a little bit stronger and be able to lift bigger weights. So again, thank you guys for the time and the attention to our research. We're really appreciative of that.
All right, thank you very much. If there are any questions, please state your name and affiliation before asking.
So it's Kath O'Driscoll from Chemistry and Industry magazine. It's a really cool technology. I'm just wondering, what are the real-world applications? I can see sort of drug delivery, but even then, is that limited a little bit by the LED, the need for this particular type of light? Are there other stimuli that could be used, and what are going to be the big applications?
That's a great question. That is one that I ask myself on a daily basis, right? And I don't mean that to say that we don't have that we don't know the answer to it, but no, we're basically. So let's start with the first example that you mentioned, drug delivery. That's a great one, right? And so one of the things that we really want to do is to be able to use near IR light to activate these gels, right? And so what that means is that you're beyond the visible wavelength now. You're past the red wavelength for light. And so that means you can penetrate tissue. And so if you think about nerve regeneration, for example, one of the biggest challenges in biomaterials is, let's say you want to embed cells in a gel. Somebody has spinal damage, right, spinal cord damage, and you want to try to regenerate that spinal cord. They actually use hydrogels where they put nerve cells in the gels, and then they inject that into that region where they want to regrow the spine. But one of the things is you have to have the gel degrade so that the cells can start to form tissue and can integrate with the damaged spinal cord. How do you get the cells to release? How can you activate that system? So one of the ideas we were thinking about is maybe using this type of technology, but have it be activated by near IR light or something that can penetrate tissue such that you can start contraction processes and release. So drug delivery is definitely something that we're very interested in, or cell delivery if you want to broaden that out. Another type of application is that we're really interested in is the photo patterning aspect, because what I didn't mention is that when we photo pattern, we actually see changes kind of in the profile of the surface of that. So if you think about, it might be good to go back. Let's see if I can just forward it and bring up that image again. Yeah, there you go. So if we see different changes in the height, then it might be possible to use this as a stamp, a reversibly photo pattern stamp for microcontact printing, right? So when you want to form self-assembled monolayers, whether it be for molecular electronics or electronics industry in general, you need to be able to stamp out precise patterns. And so if we could photo pattern with very high resolution, right, nanometer level resolution, then we might be able to make these types of, or micrometer resolution, and make these types of microcontact printing stamps. And then whenever you want to change the pattern, you don't have to make a new mold anymore. You just oxidize it and then rephoto pattern with a different pattern that you want, and then use that to then pick up molecules and try to stamp them out onto another surface. So that's one application. And then another one I'm thinking about, got a lot, I told you, mechanobiology, right? So think about when you grow and you're growing, your cells are constantly dividing, and then they bump into each other. And then eventually they start to form layers, and then those layers become organs, right, tissue. And so the question is, how does your body know how to stop that process? How do you stop growing? And that's literally the definition of cancer, right? It doesn't know how to stop. It just keeps growing, and it keeps going berserk. Well, it'd be really interesting to use these gels to maybe grow in vitro, grow tumors and things that we could use for studying cancer and the treatment of cancer. It would also be interesting just to see how different cells respond to applied mechanical stress. So if you put cells in these gels, and you start to contract these down, you apply mechanical forces to whatever's on the inside. It feels that environment, right? And that's what the healing process is about. It feels when there's certain defects in the tissue and so forth, and so then the body responds. It has this response that ends up going through these self-healing cascades. And so this would be something really interesting. So we're looking at biomedical applications, material applications. We're actually talking to a company for interest in desalination devices as well, being able to draw water across and then release, across a membrane and release it. That would be a very straightforward technology. Obviously, we need to speed it up. It's a little slow right now, so it's not quite where it needs to be for that application, but we're working on it. So this is, again, my lab's only been in existence for about 2.5 years, so we're very excited for the next 30.
Just going back to the self-healing, how does that part of it work? How do they self-heal?
Yeah, so these are not self-healing. These are just regular cross-linked, what are referred to as kind of thermosets, meaning just this absolutely cross-linked. And the only way, so if I pull hard enough, I'm going to break chemical bonds, it's not going to heal. But we are now working on programming these with the ability that when I do tear it apart, I can take the two pieces and stick them together again and then have them self-heal, quote unquote, and merge back together. And so that's something that we're actively working on as well.
Can you say anything about the chemistry of how that self-healing
There's
happens?
a lot of different types of chemistry. It's usually dynamic covalent chemistry and exchangeable bonds that can happen. So if I were to pick this up and tear it and then bring the edges back together, there are going to be dangling functional groups that could have the ability to recombine and either form covalent bonds or form host-guest complexes, or there's a number of different ways, different mechanisms by which people have been able to introduce self-healing into gels. But that's something we're very interested in, because then we could also 3D print these, right? Then I can 3D print it into whatever shape or design that I want, and I now have our active polymer in it, and it has the ability to contract that particular shape. So this is something that's very difficult when you make an object. How do you get it to, like, normally it's just a permanent thing. It's like this mouse is not going to be anything other than a mouse for the rest of its life because it was injection molded into a piece of plastic. But it'd be really flipping cool if we could actually change this and change its properties if we wanted to, right? And not be so trapped, quote unquote. So it's nice to have the ability to have stimuli responsive
So it's
materials.
like a recycling for the hydrogels then? Is it kind of that we could reuse them for all kinds of different applications?
Could be.
I mean,
Absolutely. Could
yeah.
be. Yeah.
Okay, thank you.
Yeah.
Other questions?
Yeah, we have an online question from Mr. Chaotic, who is a graduate student. He was wondering, how efficient is the return of the properties after irradiation? Is the gel just as stretchable as the virgin material, or do you only regain, say, 80% of the tensile properties?
Yeah, so that's a great question. That is something that we are literally looking at right now. What we've done is just kind of these crude tensile measurements where we clamp a gel and then we hang weights off of it, and it's almost like a what they call a creep test, where you're just looking for elongation or deformation in the gel over time. But what we've observed is when we actually measure, so we're doing the tensile measurements now, but when we use rheology, which is a way to measure kind of the stiffness of the material, we actually see that the properties are actually recovered pretty well. And so a lot of that has to do with the fact that you need two components to reverse this process. One is obviously it needs to reoxidize, and the other is you need water or some sort of solvent to actually swell it back up. And then when it swells back up, we've seen that the reversibility of the mechanical properties are actually pretty decent over the first 3 to 5 cycles. We haven't gone much further than that yet, but we're getting into doing more of those types of mechanical testing to really understand how, when we change the composition of the polymer and change how we cross-link it or integrate it into the network, how does that affect the bulk properties? In fact, that's what my NSF grant application was about. So still waiting to hear back on that one. All right.
Laura Cassidy with ACS. Do you think you could ever speed up the contraction rate enough to actually use this as an artificial muscle, like implanting into somebody who has muscle damage?
Yeah, so that is a big, big dream of ours. Again, as I mentioned, it is, so far, it's not a super fast process, so you'd be sitting around waiting quite a while if you were trying to use it. But that is something that we want to do. And one of the ways that I think there are many different possible engineering mechanisms that we could employ that would allow us to speed up the rate of the contraction process, and it also might be a little bit faster too if we were to do this electrochemically. So if you think about robotics and things of that sort, it usually requires really high voltages. These materials don't require high voltages, so if we could pump electrons in via electrochemical stimulus, that would be really nice. That's something that we're very interested in doing. We haven't done it yet. But yes, we'd like to try to speed them up. But basically what I've noticed in this field of stimuli-responsive materials and artificial molecular muscles is that the larger it gets, the slower it gets. The smaller it gets, the faster it is. So we're looking at both ends of that spectrum, right? Thinking, okay, here's large macroscopic gels, and over here is microparticles and things. So yes, that's definitely something that we're trying to do. In our recent work that'll be published, I guess, pretty soon, it's undergoing revision, we've shown that we've actually sped up the contraction process just by changing some of the linkers that in between the active units of the polymer, and it's actually a lot faster. It's like 4 or 5 times faster. So we're working towards that.
Okay, and you mentioned that you're using very small amounts of the active polymer. Can you increase the speed simply by using more, or does that not work?
We have observed that you do see some increase in the rate of performance, but then when you start to put too much in, you start to change the bulk properties of the rest of the material. And so when it's really low, it's as if this gel would have the exact same properties, essentially, whether it had our redox active polymer in it or not. And so it's minimally invasive in terms of changing and affecting the mechanical properties of the materials that you want to use, but yet it's enough present that it can cause it to contract and change mechanical properties. Yeah.
Okay, thank you.
Charles Berquist from Science Friday. So if you have your rectangle of material and you only irradiate part of it, does only that part contract, or does the reaction sort of spread throughout the rest of the material?
Yeah, so that is a really great question. So it does not spread through the rest of the material because as constructed, and as you saw with the photo pattern of the U.S. flag, it's not conductive. The material's not conductive. So when I shine light in one particular region, the electrons, because there's so little of our redox responsive polymer present, it's going to run into a non-conductive thing before it ever sees a conductive polymer, right? And so you can actually control the regions where you actually see the change, right? So if it were conductive, you're absolutely right. We could irradiate one end, and then those electrons are generated now, and then they could hop through the material, and then you would see kind of this gradient change, which would also be really cool. It's something we're also looking at too, in terms of trying to make them conductive. But for this particular construct or this design, where it doesn't happen because it's not conductive. So wherever you introduce the electrons, there's a photocatalyst that absorbs the visible light and it transfers an electron to the polymer. That's it. That's where it stops right there. It doesn't propagate through the material.
And second, the speed of the recovery mechanism. Once you want to oxidize it back, how quickly is that process done?
So the oxidation, if you just do it like this, where with the flag, where it was not put in any type of solution or anything, I think that takes approximately 60 minutes to go from photo pattern to all the way back to blank slate now. But if you do this in water, you get swelling and diffusion and things that happen more quickly. And so the process can be sped up a little bit, but it's still on the order of tens of minutes for the full oxidation. Because, think about a dried out sponge, right? Which is a kind of a three-dimensional network. When it dries out, it's smaller. Then you drop that in water, and it kind of swells up, and those polymer chains swell up, and that's the water that's kind of filling that void space. Same thing happens in a hydrogel, right? Like I said, 5% of this is polymer, and the rest of it's water, so it's swelling. If we could speed up the contraction process, then we'd like to also speed up the expansion process as well. Yeah.
Other questions?
Yeah, we have an online question from someone going by Monkey.
All right. Monkey squared, what's up?
Yeah, so they're asking, does the frequency of light act as a gradient of change too?
Man, these are great questions. This is something we are also looking at in terms of, I love thinking about ideas constantly about this stuff and how we can activate it. We are looking at different intensities of light. We are looking at different wavelengths of light. As I mentioned, for biomedical applications, it would be nice if we could go more towards red light or near IR light, and that's something that we're actively pursuing. And that is actually one of the questions I have is the wavelength of the light and whether it's direct irradiation or if it's a gradient where the gel is tilted and then you're shining light here, and so the things closer to the surface are actually being irradiated first versus the things that are closer to the light are being irradiated first versus the things that are farther away. These are great questions. We want to see if we can not only have different wavelengths of light, but control the contraction process in an anisotropic manner, meaning it's not all shrinking in every dimension at the exact same rate. So that is something we're also very interested in. Yep.
All right, thank you very much. This has been a fascinating session. The archived version of the session will soon be posted at bit.ly/ACSlive_Orlando 2019. Please join us for our next press conference at 1 p.m. today on new alternatives that may ease demand for scarce rare earth permanent magnets. Thank you very much.